Hi everyone, I'm Alejandra and I'm super excited to be here with you today in this first edition ever of Virtual Be a Scientist program. So I'm going to start first telling you a little bit about myself.
I'm from Uruguay, a very small country in South America. Last summer, I had a unique and amazing opportunity to move to Berkeley and start doing experiments in the university. I work with proteins, which are molecules produced by cells. There are thousands of different proteins. Every living organism has its own set, even you and me, and this set changes over time and in different conditions. Each protein has a specific function and lots of them remain unknown, which is super exciting as there's still a lot to investigate. Some pathogens have evolved to have proteins that help them to survive and infect others. It's like superpowers they have. My work in Uruguay and now in Berkeley focuses on studying these proteins to try to understand how they work. It's been discovered that proteins can have very useful applications. This is now called biotechnology, but in fact it's something that exists and has been used since ancient times. One cool example is the use of the yeast Saccharomyces cerevisiae, which has a set of proteins that helps it to consume sugar and produce useful products for us.
So now I have a science challenge for you. We are going to do a short experiment where we are going to evaluate whether the yeast is able to use sugar or other ingredients as a nutrient, and also we are going to see which is the outcome. So first, if this was the lab, my kitchen, the first thing that I would do is put my lab coat. As I don't have the lab coat here, of course, instead I'm going to use this kitchen apron as a protective personal element that is very important to use when you're doing experiments. And also, if you have long hair, you need to tie your hair. That's also very important. If we were working with hazardous chemicals, we would need to use gloves or glasses, but in this case, it's not necessary. So let's start. So you need to get your materials and your ingredients first. So here you can see the things that we are going to need are 1 teaspoon, 1 tablespoon, 1 measuring cup, a Sharpie or a pen to label, and also you're going to need 3 Ziploc bags. These bags need to be hermetic, so you will need to test them maybe first with water or something because they don't have to lose any gas. I'm going to use only 2, but you're going to use 3. Now you're going to see why. Also, you're going to need yeast, sugar, and other ingredients that you're going to choose. Besides, you're also going to need a clock or something to measure time, and also a pen and a notebook to write all your observations. So here, for example, I started putting that this is going to be called the yeast experiment, and we are going to annotate all the materials and regions, and we are also going to annotate how we set up the experiment, and then we are going to write our observations. So let's start. How are we going to do it? We are going to put in each bag half a teaspoon of yeast. And remember that you're going to have 3, so you're going to put in a third one, also half a teaspoon of yeast. This is a active dry yeast that I'm using. And then we are going to label one of the bags as sugar. The other one is going to be negative control. And then only the one with the sugar, we are going to add 1 tablespoon of sugar to the bag. In the third bag that you're going to have, instead of adding 1 tablespoon of sugar, you're going to add a tablespoon of other ingredient. That could be anything that you have that you usually use as food in your kitchen. It could be, for example, I don't know, honey or some fruit or whatever you want. Then we are going to mix it very well to have like an homogeneous solution. This one doesn't have anything. And now we are going to add the water. The water we are going to measure with the measuring cup. Needs to be warm. It doesn't have to be super hot because we are going to kill the yeast. And it doesn't have to be cold because yeast also don't like cold water. So it has to be warm. How do we know if it's warm enough? It's just something that we touch and we feel like nice. Okay, so I'm going to open here, and when I feel like the temperature is nice for me, a little bit more. I'm going to put like more or less 20 milliliters of water. Then I'm going to try to take all the air that is inside the bag maybe you'll need to put it in the bench. Try to take as much as air as you can, and then you close it. Then you mix it very well until you see that there are not pieces of yeast together. So after we have all our bags ready, remember that you're going to have 3, we put them over a cloth and we put it like this. We set our time to start.
So here I show you my results after 1 hour. What can you conclude about the results? What are some common uses of yeast? Can you find other proteins with useful applications for us?
So to finish, I hope that you have fun with the experiment, and I'm very excited to see your results. So see you soon and have fun. Bye!